In this video, we shall discuss the physiology of the thyroid gland. Let us assume that this is a follicular cell of the thyroid gland. This part of the cell faces the lumen of the thyroid follicle, which is filled with colloid. So this is the luminal aspect of the cell, and that is the basolateral aspect of the cell. Like most cells in the body, this cell has a nucleus and cytoplasm. The nucleus contains DNA, and the DNA contains various genes, and these genes, upon transcription and translation, produce various proteins. For the purpose of this discussion, we shall consider three proteins. The first one over here, as you can see, consists of a large number of amino acids marked in blue. The amino acids marked in green are tyrosine residues. Now this protein is called thyroglobulin, and it is seen inside the lumen together with the colloid. The second protein that we shall consider is an enzyme called thyroid peroxidase enzyme, TPO, and it is seen on the luminal aspect of the follicular cell. The third protein is seen on the basolateral aspect of the cell, and it is called the sodium iodide symporter. Now, whenever iodide circulates in the circulation, it is brought to the thyroid follicular cells, and it enters the thyroid follicular cell via the sodium iodide symporter. As it moves towards the luminal aspect, it encounters thyroid peroxidase enzyme. Now this enzyme will convert iodide in its ionic form into reactive iodine species. Now this process is what we call oxidation. Now the reactive iodine species can be added to the tyrosine residues of thyroglobulin. So two molecules of reactive iodine species can be added to one molecule of tyrosine, or indeed one reactive iodine species can be added to one tyrosine residue of thyroglobulin. This process is called organification. It is also carried out by thyroid peroxidase enzyme. The result of organification is the formation of diiodotyrosine or monoiodotyrosine. The next thing that occurs is that adjacent molecules of diiodotyrosine can be linked together. Similarly, a molecule of diiodotyrosine can be linked to an adjacent molecule of monoiodotyrosine. This process is called coupling, and it is also a function of thyroid peroxidase enzyme. The result of coupling is, of course, the formation of T4 and T3. Hence, the enzyme thyroid peroxidase is responsible for carrying out all these three actions, namely oxidation, organification, and coupling. We know that the anterior pituitary secretes TSH, and it can bind to its receptors on the follicular cell of the thyroid. And when this happens, there is endocytosis of the colloid, which exists in the lumen of the thyroid follicle. The colloid gets endocytosed into the follicular cell, and as this happens, thyroglobulin is also endocytosed. It is protected in lysosomes, where it undergoes proteolysis, and T4 and T3 are released into the circulation. The predominant product which is released into the circulation will be T4, which is the less active form of thyroid hormone. Now, within circulation, thyroid hormones are carried in association with carrier proteins. There are many carrier proteins, but the most significant among them all is thyroxine binding globulin, or TBG. So once thyroid hormones, predominantly T4, is carried in the circulation to all parts of the body, it will enter the target cells. Now the target cells, like most cells, have got nucleus and cytoplasm, and the nucleus contains DNA. Now in the absence of thyroid hormone, that is in the unliganded state, the thyroid receptor exists within the nucleus, bound to the DNA, to certain specific parts of the DNA called thyroid response elements. Again, in the unliganded state, it is associated with certain proteins known as co-repressor proteins. So this is the situation in the unliganded state. Now, other parts of the DNA contain genes which upon transcription and translation produce proteins, a group of proteins known as intracellular deiodinases. When thyroid hormone, when T4 enters these cells, it is converted by these intracellular deiodinases into T3. T3 enters the nucleus and displaces or reduces the affinity of co-repressors. The co-repressor is replaced by co-activators. So the conformational change that T3 produces makes the affinity of the thyroid receptor much more towards the co-activators. So now these three form a complex: thyroid receptor, T3, and the co-activators. They will now stimulate the thyroid response elements to produce transcription, translation, and they produce the proteins which carry out the various actions of thyroid hormone. Now, once its action is complete in the cell, T3 will dissociate from its receptor and returns into circulation. It finally reaches the liver, where it undergoes conjugation by glucuronidation and sulfation, and these conjugates enter the gut. Now, once in the gut, two events may occur. One, the thyroid hormones remain conjugated and get excreted in the feces. Secondly, deconjugation can occur in the gut, and the deconjugated forms can enter portal circulation and re-enter the liver in a process known as enterohepatic recirculation of thyroid hormones. During this process, some amount of thyroid hormones can escape enterohepatic recirculation and can enter systemic circulation again and go on to stimulate thyroid receptors in various parts of the body.